need a year to a year and a half. Chinese firm Sinovac is meanwhile creating an inactivated vaccine. Using this more than 100-year-old method, the viruses are simply killed and introduced into the body. The danger here is that the immune system can overreact so much that it kills the person. But experiments on mice and monkeys have shown that this vaccine can be a very successful and effective solution. It's now being tested on humans as well. The advantage of an inactivated vaccine is that it's extremely simple to produce and can be done in almost any country. A team at UK's Oxford University is hoping to have a vaccine ready by September 2020. They've opted for a third method, live vaccines with viral vectors. This method uses a different, harmless virus as a vehicle. It's genetically modified so that the surface imitates the typical structure of the coronavirus, its spikes. When the virus, disguised in this way, is administered, the immune system forms antibodies that would also fight the real COVID-19. When the virus, disguised in this way, is administered. Although no one knows if the UK's active ingredient even works yet, a company in India has already started mass producing it.
I'm joined now by internationally renowned medical researcher Sir Jeremy Farrar. So very good to have you on the show. Starting on an optimistic note, when do you expect we'll have a vaccine ready?
Being optimistic, I would say we'll have a vaccine that we believe to be safe and which we're starting to see evidence that we hope would be protective. We would have that in the fourth quarter of 2020. But that's only the start, because having a vaccine at that stage is not enough to vaccinate everybody all over the world, and that's where we need to work to. So that is going to take a few months later, and we need to do all of these things in parallel rather than all in sequence to get there as quickly as we can to offer vaccines to the world.
Right. You mentioned a very sensitive point there, because let's say that we do get a vaccine this year. Who's going to be in charge of distributing it, and how can we ensure that that will be fair?
That's a great question, and of course it has to be fair. We have to believe in a world which is equitable and which access to this vaccine, because this pandemic is affecting the whole world, that this access to any vaccine is not determined by any of our abilities to pay for it. It isn't right, it's not fair, it's not equitable, it's not the sort of world we want to live in. So making a vaccine available to everybody, wherever they are, is the crucial element here, and pay tribute to the German government in this, in the support for CEPI, which is driving this process, but also the World Health Organization, who have made it very clear that having a vaccine for the rich world is not enough, and it's not good enough. We have to commit to having a vaccine for the world.
Right. And the European Union certainly says that we need a global response to the pandemic. There's even an event coming up next week. How do we get everyone on the same page?
It's difficult to get everybody on the same page. It's difficult at the best of times, but of course, as everybody knows, we're living in very tense geopolitical times at the moment. Again, the European values, the values of solidarity and the values of equity and the values of a sense of fairness are absolutely critical here. And the role the European Commission has played, alongside Germany, alongside France, alongside many of the European nations, the UK as well, has very strongly argued for not just the science, not just the development, not just the manufacturing of this vaccines, but that we're absolutely committed to making this available globally. And the leadership from the European Commission on this has been the reason we're going to be having this pledging conference next week. It would not have happened without that leadership.
Right, and it's also, of course, about funding. You yourself say that the world needs at least $8 billion to get the crisis under control. Now, we've seen huge stimulus packages aimed to boost economies worth trillions of dollars. So $8 billion can't be such a big problem, can it?
It isn't. I mean, the latest figure I heard at the weekend when joining a group of economists and people from Federal Reserves and central banks was that the world is losing somewhere in the region of about 200 billion dollars a week Just play that out across the whole world and play it out for just the 3 or 4 months that we've already been through, let alone the continued disruption going forward. The investment of $8 billion now, which is just the start, that will not get us to a vaccine to everybody in the world, but it will get us to the next critical stage, which is doing the science, the research, and the manufacturing of those early vaccines. It is a tiny amount of money. I cannot imagine a better investment at the moment than putting that money into science in order to develop. It is a tiny amount of money. I cannot imagine a better investment at the moment than putting that money into science in order to develop. Concentrating on vaccines. It's vaccines, it's diagnostics, and it's also drugs and therapeutics. We need all three. I was a junior doctor at the start of the HIV epidemic, and many people talked in the late 1980s about how a vaccine for HIV was going to change the world, and indeed it would. But some 30 or 40 years later, we don't have a vaccine for HIV. Indeed, we don't have a vaccine for tuberculosis or malaria either. And yet, actually, through good diagnostics, through good health systems, through good public health, and through having good drugs, we've been able to keep the horrible pandemics of HIV, TB, and malaria. Although we haven't finished those pandemics, we have made great progress in bringing them under control. So there is no single magic bullet here. It's not all about vaccines. It's not all about diagnostics. It's not all about drugs. It's about all of those, and also social sciences that brings it all together. We would be unwise to put everything into the vaccine camp and not also produce the drugs and diagnostics that are required.
That's Jeremy Farrar there. Thank you so much for your time and for your input.
Thank you very much.
Time for some of your questions now, and today focusing on animals. Here's our science editor, Dave Williams.
Can the coronavirus spread in animals?
Yes. Coronaviruses are what are sometimes described as being pretty promiscuous, and there's every indication that the showed that, and the evidence was backed up by the news that a number of lions and tigers at the Bronx Zoo in New York had contracted the disease, probably from an asymptomatic zookeeper. It's clear that we can give the virus to other animals, but it's not yet clear whether or not at least our domesticated animals can give it to us.
What should I do first when I come back from walking my dog? I can't bathe him daily.
In previous epidemics involving related coronaviruses like SARS, dogs owned by people who had become sick didn't grow sick themselves or transmit the virus to other humans. That seems to be holding true for COVID-19 as well. As to the question of whether SARS-CoV-2 could stick to your dog's fur, that also appears to be fairly low risk as hair is porous, which will tend to absorb and trap any virus. That said, when you're outside with your animal, the same social distancing rules should really apply to them as apply to us.
Can I use an ultraviolet lamp to sanitize a room? What effects would that have on my body?
Ultraviolet, or UV, is the short wavelength invisible radiation emitted by the sun that causes sunburn, and after prolonged exposure can cause skin cancer. So high doses of it are dangerous to living things and also to viruses. We can make lights that emit those specific wavelengths, and they're often used in hospitals or labs under controlled conditions to kill microbes in the air and on surfaces. There are companies now selling devices for decontamination in the home, but experts at the WHO and elsewhere have warned against using them, especially when it comes to trying to sanitize, for example, your hands through direct exposure to UV light. The problem is basically that in order to damage any virus, the radiation would also have to be intense enough to damage you. UV radiation is a potentially dangerous tool, and trying to use it in the home is a lot more likely to do harm than good.
Now, until there's a vaccine, social distancing is key to keeping the pandemic under control, but that's tough around national holidays. So the Swedish city of Lund came up with a creative measure. Authorities covered the Central Park with chicken dung. Tens of thousands of Swedes usually gather there to celebrate Walpurgis Night, but not this year, after the council spread a ton of the stinking manure on the lawns. The perfect deterrent.
Welcome back to the update 4 in the series of COVID-19. And in this lecture, I will be talking about the vaccine candidate being developed by the scientists at the University of Oxford's Jenner Institute and Oxford Vaccine Group. And in this lecture, I try to give you the complete story that surrounds this vaccine. And my dear friends, before I start this lecture, let me give you the outline of this lecture. See, firstly, we will be talking about the story of preclinical development of COVID-19 vaccine, which is being carried out by the University of Oxford's Jenner Institute. And next, I will be focusing on the current status of clinical development of vaccine. Next, I will be talking about the timeline for the completion of these trials and when it's going to start its production. And lastly, the special part of this lecture where I'm going to talk about the connection between Serum Institute of India and the University of Oxford's Jenner Institute. You know that many scientists around the world are working so hard to develop a vaccine to prevent and cure COVID-19, right? And according to news portals, on 10th January 2020, a team in the University of Oxford's Jenner Institute led by Professor Sarah Gilbert, Professor Andrew Pollard, Professor Teresa Lamb, Dr. Sandy Douglas, and Professor Adrian Hill, they started their work to design a vaccine candidate. In a statement issued on April 23rd, the University of Oxford said they have made a vaccine candidate called CHAdOX1 novel coronavirus 19. And they explained the way how they developed this vaccine CHAdOX1. They said that firstly they have taken this adenovirus that causes common cold infections in chimpanzees, right? I know you are thinking why only chimpanzees. Guys, we humans share 99% of our DNA with chimpanzees and bonobos. Secondly, they said that this adenovirus which have been taken from chimpanzees have genetically altered by adding the genetic material of spike glycoprotein of SARS coronavirus 2. And they named this whole thing as chimpanzees adenovirus OX1 vaccine viral vector. So that's what the name CHAdOX1 novel coronavirus 19, where CH is for chimpanzees, AD is for adenovirus. I think OX1 is due to because they have genetically altered this adenovirus genome, so they have placed it like OX1, and it's a vaccine as well as it's a vector because you have been modifying this genome and it's a viral vector, right? They have made this vaccine candidate in such a way that after giving this vaccine to a human, it is impossible for the modified adenovirus to grow in humans, right? So before proceeding further, let me tell you briefly about the spike protein. So it's usually found on the surface of SARS coronavirus 2, and this spike protein, it plays an essential role in the infection pathway of the SARS coronavirus 2. And this SARS coronavirus 2, what it does is it uses this spike protein to bind to a very special type of a receptor that present on human cells called ACE2 receptors. So by binding with these ACE2 receptors, the SARS coronavirus 2 is going to enter in the human cells, thereby it infects that human cell. So it's nothing but the spike protein is like a priority pass where you need to show to the human cell ACE receptor before you want to enter into that cell. So once the pass is verified, then you are going to enter into the cell and you are going to cause the infection. And this is advantage of having spike protein for this SARS coronavirus 2. So as to stop this entry, we have introduced the genetic material of the spike protein in adenovirus genome to make a vaccine like chimpanzees adenovirus OX1 novel coronavirus 19, right? And when you give that candidate vaccine to humans, this will help the human body to recognize this, and this will help the human immune system to make antibodies against the spike proteins. And these antibodies that were produced, they are going to mount an immune response on these spike proteins, and they are going to prevent this virus from entering the human cells and causing an infection. Followed by, in the same statement issued on April 23rd, Oxford University also mentions that they have initiated a phase 1 human clinical trial of its vaccine CHADOX1 novel coronavirus 19 against the SARS coronavirus 2, right? You know, before going to clinical trials, either it's a drug or vaccine, it needs to prove its safety in animals. Here the question arises, has the vaccine is tested in animals? The answer is yes. The university had the past clinical trials data where they have used this adenovirus construct when the vaccine was being developed as an option against MERS. That's the reason the vaccine leapt straight to human trials for testing of its efficacy. So what is a preclinical development? See, preclinical development is a stage of drug development or vaccine development process that begins before clinical trials, during which experiments on efficacy, pharmacokinetics, safety pharmacology studies, toxicity evaluation studies are carried out. And this development process aims at determining the safety dose for humans and assess product's safety profile. This is the topmost priority of having preclinical studies, knowing the safety profile of your drug or of your vaccine, right? And my dear friends, in this case of COVID-19 vaccine, we have done the major part of preclinical studies before, when we are developing the adenovirus construct to be used to develop a vaccine against MERS. So it becomes easier to directly jump onto the human clinical trials, right? So after the preclinical development of vaccine, we proceed to clinical development of a vaccine, right? And the successful clinical development would lead to clinical approval of vaccine, which is ready to be introduced in the market. See, basically we have 4 phases: phase 1, phase 2, phase 3 and phase 4. So now let's look at the process of clinical trials being carried out by the Oxford University. So you can put a broad heading: phase 1 single-blinded randomized placebo-controlled trials. See, what do you mean by single-blinded? Nothing but only the researcher doing the study knows which treatment or intervention the participant is receiving until the trial is over. A single blind study makes results of this study less likely to be biased. And whereas when you see the placebo-controlled trials, it's a term which is used to describe a method of research in which an inactive substance, or you call it as placebo, is given to one group of participants, right? This is the first part of that word placebo, followed by the treatment, which is usually maybe a drug or vaccine, is being tested, is given to another group. So it's a control. So you're having a placebo as well as you're having a controlled. So that's what we called placebo controlled trial. So here in this clinical process, up to 1,112 healthy volunteers from Oxford, Southampton, London and Bristol have been recruited for the phase 1 trial. And these volunteers involves both males and females aged between 18 to 55 years. All the participants or volunteers will be randomly assigned, or they split into 2 arms, the vaccine arm and the control arm. In this case, in the control arm, the control will not be a placebo as is usually done, but here we give another vaccine. So this will be a vaccine called MenACWY, and this is a vaccine which is licensed for use against a common bacteria that causes meningitis and sepsis. Whereas the other arm, called vaccine arm, is going to receive the candidate vaccine called CHADOX1 novel coronavirus 19 for comparison. And you are thinking why the Oxford University is using MenACWY vaccine rather using a placebo or a saline. See, this is being done because CHADOX1 nCoV-19 vaccine causes some side effects such as headache and fever. So using this placebo such as a saline control would not produce those same side effects. Thus the subject would know what he or she had received. So they have used this ACWY vaccine, rather giving simply a saline. And the participants will not know if he or she received a candidate vaccine or not. That is why it's a single blinded study. And so as to assess the dosage and immune response of the candidate vaccine, the university researchers will also be testing 2 doses of the candidate vaccine given 4 weeks apart on a small group of 10 volunteers, right? So moving to the next part, what is the timeline for the trial? See, the phase 1 trial is expected to be completed in end of the May if transmission remains high in the community, followed by the phase 2 trials may be completed by August to September. And according to Suresh Jadhav, who is an executive director of the Pune-based Serum Institute of India Private Limited, phase 2 and phase 3 trials may get combined if the phase 1 trial results are encouraging, right? Now the last special part of this lecture, talking about the connection between the Serum Institute of India as well as the Oxford University. Serum Institute of India will be now the biggest manufacturer of this vaccine to the entire world. See, the Serum Institute of India, one of the largest vaccine manufacturers in the world, is being partnered for the production of vaccine in preparation for the trials. According to Mr. Jadhav, as I said earlier, he is an executive director of Serum Institute of India Private Limited. He said that the company will start manufacturing the vaccine the moment the phase 3 trial or the combined phase 2 or phase 3 trial begins. So if the last 2 stages of the trial are combined, then it would start manufacturing the vaccine by the end of June, and it would be ready with millions of doses by the end of this year. And the company is saying that it is so confident of manufacturing 60 to 70 million vaccine doses by the end of this year. So my dear friends, this is my complete update on vaccine candidate that is being undergoing clinical trials at Oxford University. If you like this lecture, please do share this lecture with your friends, and we meet in the next lecture with a new update or concept. Till then, stay tuned. Have a nice day.